Hey Kartik, what are you doing?
Nothing.
Hey, come on, let me see.
Oh. Alright, fine.
The original clip of Jurassic Park.
DNA strand, like me, is a blueprint for building a living thing. And sometimes animals that went extinct millions of years ago, like dinosaurs, left their blueprints behind for us to find. We just had to know where to look. 100 million years ago, there were mosquitoes, just like today. And just like today, they fed on the blood of animals, even dinosaurs. Sometimes, after biting a dinosaur, the mosquito would land on the branch of a tree and get stuck in the sap. After a long time, the tree sap would get hard and become fossilized, just like a dinosaur bone, preserving the mosquito inside. This fossilized tree sap, which we call amber, waited for millions of years with the mosquito inside, until Jurassic Park scientists came along. Using sophisticated techniques, they extract the preserved blood from the mosquito and, bingo, dino DNA.
Movies, fact or fiction, Jurassic Park edition.
Of movies, fact or fiction. Today, the movie we'll be talking about is a very interesting Jurassic Park. Now, we have invited 3 very interesting scientists to tell whether this movie is correct or wrong. So how are you guys doing today?
Good.
Yeah, we're doing good.
Great.
So, would you guys like to explain to us what's wrong with the movie?
Well, many of the procedures taken throughout the movie are not scientifically correct, especially the process taken to make the dinosaurs in the theme park. Part in the movie specifically is wrong. The way they did it was they extracted dinosaur DNA from the mosquito preserved by amber. However, the procedure taken to do it has many flaws to it.
Really? So could you explain as to what some of the flaws are? So, there was a small part of the movie that showed the Jurassic Park scientists drilling a hole through the amber to get to the mosquito, then putting a thin syringe in and extracting mosquito blood.
And the mosquito blood contained dinosaur DNA. The reality of this is not very high because the scientists drilling in the hole could have drilled other DNA particles into the mosquito. When you are drilling something, you are allowing any DNA particle in the air to enter the mosquito.
Correct, and the amber itself contains other microscopic bits of things from the past that have their own specific set of DNA. When drilling and using a needle to extract the DNA, you have the chance to pick up any other DNA, not of dinosaurs.
Which is why we have prepared our own little movie that uses the correct procedure for extracting the DNA.
Okay then, let's roll the film.
The real way of mosquito DNA extraction. First, we have to cut around the circumference of the amber by using a circular saw. We have to stop short of the mosquito so we do not cut any of the insect apart. Next, we take the small piece of amber and dump it in liquid nitrogen so it becomes brittle enough for us to crack. After this, we take the nugget into a clear plexiglass box that has positive airflow so any contaminants will not get inside and dirty the amber, and ultraviolet light to shine on to disrupt any other DNA. Now we have to pull apart the mosquito and put each of its fragments in separate test tubes. The test tubes are then sealed tightly so nothing gets into them. We then move into another lab with a clean environment and use another plexiglass box with plenty of ultraviolet light to disrupt DNA and positive airflow. After we have moved, we take a soapy solution and another substance to dissolve proteins and lipids of the cell membrane. Phenol is the next thing to add as it will separate the DNA from other things in the mixture. The DNA molecules will end up floating around in the water from the soapy solution, and the phenol will have dissolved most of the fatty structures of the cell membrane. Next, we spin the test tubes on a centrifuge to separate the solutions. After spinning it on the centrifuge, we use a pipette to extract some of the water-containing DNA. Now we add cesium chloride, which is almost like sodium chloride or table salt. We then take the new solution and spin it on an ultra centrifuge to make the cesium chloride denser at the bottom, and the DNA will gravitate to the level of a single layer. We then add a dye that is designed to attach to DNA molecules and to signal where they are in the tube. The DNA will then have appeared in a red glowing color, and we will have dinosaur DNA.
Okay, you want this one?
Dude, sugar is the best, the original best.
What's we get 2 each? I want this.
Hey, what's that one?
Why are you videotaping? Oh my, stop it.
Because it has to...
We're gonna start, go. Hey, shut up!